All right, welcome to this viticulture session for the Oregon Wine Symposium. So today we'll talk about sap flow pruning and that will prevent trunk disease. I'm extremely excited to be part of this. I'm actually more than excited, and you're going to understand why. So first, I would like to introduce two speakers today. Dr. Kendra Baumgartner from California, a USDA researcher focusing on disease epidemiology and focusing on trunk disease, and Mia Fisher from Simon-Sir, sorry, you see, I didn't really do well, who is basically a consulting company, very internationally renowned, that will teach, preach, and help viticulturists to get the best techniques for pruning, and especially against trunk disease. But they will introduce themselves and talk more about it. So first off, you probably wonder why a winemaker from Oregon will talk about viticulture, and you should wonder why. And I have exactly 3 minutes and 45 seconds to explain you why. So when I decided to study viticulture, about a book, and my first book was about disease in the vineyards and vines, and by turning the pages, I realized that trunk disease were extremely how can I say that, problematic, and it was something blew my mind. Some of the author was saying that one of the disease called ESCA, ESCA, was one of the most complex, the oldest and deadly disease that can touch vines and vineyards. So complex, okay, you understand there is a lot of fungi, and we're going to talk about it. That's a lot of fungi that will actually be responsible for this disease. Oldest, I trust the author at this point, and then deadly. It was like, really, it doesn't make sense to me. I never really heard about it. 2008 Burgundy, people talk about like disease, about downy mildew, powdery mildew, but trunk disease, never heard of it. So I like challenges, and I decided to go to Bordeaux and do my bachelor in viticulture at the engineering school and study in the lab of the trunk disease lab. And it was fascinating. I studied the spatial dynamic of the disease ESCA. And then another realistic thing happened to me. I realized that we have Bordeaux was extremely touched by the disease, touched to an extent that was very concerning at many point of view. Number one, I realized that the research was not agreeing on the disease. Unfortunately, it was very complicated to have different labs in France to talk about the same thing. What way can mitigate the problem, and how we can eventually, like the industry can have a good evolution with this disease. That was number one. Number two, there was no curative treatment for this disease that would be a benefit for stopping this disease. And finally, the deadly, that's when I figured out the deadly part of it. The deadly part was absolutely incredible. I visited some vineyard where vines, vineyards, entire vineyard, 30-40% of the vineyard was dead, and symptoms of trunk disease were seen, and it was very concerning. And so this is where I took it very seriously, and I said, this is a big problem. I also realized that some vineyards are touched differently, some regions are touched differently. It's different with climate, different with varietals, cultivar, farming practices. What we found is farming practice is very important for the control of this disease. And as you know now, being part of the winemaking team and winemaker at Lingua Franca, I was very fortunate to get trusted by Larry Stone to give me the hand of his 66-acre vineyard that was at the time 2 years old. And with all my background with the research on trunk disease, I was like, we need to think about this. It's maybe not there yet, but it's actually here, and we have to think about it. We have to work towards it. So we decided to convert the vineyard, or not even convert, work this vineyard with a team into a specific pruning system. And this is why I'm here. It's because I've been bugging everybody to talk about it for so long, and I'm extremely excited. to talk about this with everybody. So to finish this, I want to say that the trunk disease are the grape trunk disease that we're going to talk about today are insidious, dangerous, and they are in Oregon. You want it or not, they are here. We maybe don't see it, but I urge you to listen to these two wonderful women that will talk about it and explain you more the pathogen and the technique that can help to prevent this disease to grow.
Very good. Well, thank you so much for the invitation. It's great to be here, albeit via the computer only. Okay, so I'm going to talk today about the different trunk diseases that are found in Oregon, how they spread, and a little bit about their management. Um, as you just heard, these fungi are insidious. Um, they're hard to see because they cause internal wood infections. That said, they have symptoms that eventually are apparent from the canopy. So first we're going to talk about what goes on inside the wood, then a little bit about how to recognize their symptoms and how they spread. These symptoms that are caused in the wood by different trunk pathogens represent dead parts of the vascular system. This discolored wood you see here is dead. That said, it doesn't spread throughout the entire vine. The vines have a way of kind of limiting the spread of these infections so that they are somewhat localized near the entry point. The fungi live in the wood around that dead, discolored section. And some of them eventually make their way out to the bark surface to make spores and then potentially spread to other vines in your vineyard. We found over the years that because these infections are chronic and they're in the permanent woody structure of the vine, that prevention seems to be the most effective approach. There's no material known to science that you can spray on the outside of the vine or apply to the root system that gets taken up that will kill off these infections. Two of the diseases that are found in Oregon, which are widespread also in California, are Botryopseuria dieback and Eutypa dieback. These diseases kill fruiting positions. And so you can imagine what their impacts on yields are. Because vines are missing more and more shoots over time, they have a lower photosynthetic capacity, and that means they will gradually decline in vine capacity over time. The symptoms of these two diseases often first appear between years 6 and 8. That said, the infections are established months to years before that. The time between infection and when symptoms become noticeable due to these trunk diseases, to all of them, but especially Eutypa dieback and ESCA, which I'll talk about in a minute, it can be 3, 4, 5 years before you see noticeable symptoms after the point of infection. In susceptible vineyards, in susceptible cultivars with very aggressive trunk pathogens, the proportion of symptomatic vines can greatly increase between years 10 and 15. Again, these infections are established years before, but in year 10, the combination of a susceptible cultivar and a very aggressive pathogen might mean that in year 10, you might only see 20% of the vines showing symptoms, but by year 15, it could be as high as 75% of the vines in the vineyard showing some symptom of a trunk disease. ESCA is another trunk disease that's a little bit different in terms of its impacts on vines. Typically, with typical ESCA symptom development, the main impact is on fruit ripening and fruit development. None of the standard measures of fruit quality kind of develop in the vineyard. You'll have bad measures of Brix, TA, and that'll end up impacting phenolics in the wine as well. Because this fungus or the fungi that cause ESCA impact the photosynthetic capacity of the vine, you generally get a decline in vine capacity that's somewhat gradual over time. The symptoms of ESCA can become apparent a little bit earlier sometimes than those of the dieback type trunk diseases, maybe in years 4 or 6. That said, the infections happen years beforehand. The best time to look in the vineyard for the symptoms of Botrytis dieback are sometime between bud break and bloom. And I have a vine pictured here. And I show, for all of my photographs, I show when I took the picture during the year, the cultivar, and where I took that picture from. So you can kind of get a sense of where we're moving along in the growing season here. Um, Dead spurs are the most common symptom of Botrytis dieback. And the same is true for U-type of dieback. Also here you see pictured a stunted shoot. The Botrytis dieback symptoms don't tend to cause the leaves to be anything but green and maybe stunted. But that whole shoot will, this shoot here, pictured here, won't have any flowers on it and obviously no fruit. Eutypa dieback is a little bit different from Botrytis dieback in that it will kill spurs. It will stunt the shoots that come out of infected spurs. But the leaves on those shoots are affected by toxins that the fungus makes where it resides in the wood. They get carried out to the leaves and they cause the leaves to be shaped very oddly. They can have a cupped shape. They can look kind of elongate as if all the veins are growing parallel to each other instead of fanned out. And they can have these brown, kind of tattered edges to them. And this is all kind of thought to be due to the toxins, which disrupt hormone production for the plant. The auxins, the cytokinins aren't quite in the right balance. And so you get this odd growth, which is much like sometimes what you see with phenoxy herbicide damage. Eventually, those stunted shoots will die back. It could happen in the same growing season and it can happen in successive growing seasons. And if you cut through those, you'll see the wood cankers. This rotted, dead portion of the wood is discolored because the vine makes phenolic compounds to try to limit the spread of the pathogens. Some of the pathogens are really clever. They figured out ways to detoxify those toxins. And some of them have figured out ways to even feed off of them. That discoloration in the canker also comes from these fungi can break down the wood and feed off of the main components of the wood too. Esca has very distinct leaf symptoms, and these are best seen typically between veraison and harvest. They can appear as early as July, though, and they'll worsen through harvest. They are extremely variable from vineyard to vineyard, but they all include some amount of scorching around the edges or in between the veins, and some amount of discoloration. It can be yellow, as in the case of the Sauvignon Blanc pictured here. Sauvignon Blanc is a very susceptible cultivar to Esca worldwide. Here in this Muscat, you see a little bit of scorching, a little bit of yellow discoloration. And here in Chardonnay, we have very little scorching, but a fair amount of red and yellow discoloration. These symptoms will be associated with individual shoots on the vine, certain fruiting positions. They can vary a bit from year to year in severity, but the fungi don't go away. They're always inside the wood. As I said earlier, Esca has a pretty significant impact on fruit quality and fruit ripening. Clusters on shoots with leaf symptoms will never quite ripen up properly. They can be visibly not ripe. Even ones that are visibly ripe will be off the mark in their fruit quality parameters as well. One of the most severe symptoms of Esca, which is albeit one of the most rare ones, are the fruit spots. This is where Esca gets its common name of measles from. These are not fruiting structures of the pathogen. They are spots that are thought to form due to toxins that the fungi make where they reside in the wood that get carried out to the shoots. And fruit like this has to be dropped. Obviously, it's visibly not looking healthy, and the berries crack, and also they never ripen up properly. This is a big problem for our table grape industry in California, where even though the fruit is picked with very low bricks, if it has any sort of blemish on it, it can't be sold. The wood symptoms of Esca are different from those of the dieback type pathogens. You don't get cankers, but instead you get, depending on the pathogen that's present causing ESCA in your vineyard, you get black or dark brown spots. There's spots in cross section. If you look at them lengthwise, they look like lines. This sample here is from Oregon. This was sent to me by an Oregon grower. It's Pinot Noir. And then another set of ESCA pathogens, which are actually wood decay fungi that cause white rots and brown rots of wood. They cause a wood decay. And depending on the pathogens that are present in your vineyard, you may see one or both of these symptoms, sometimes in the same vine. ESCA can also cause a very rapid vine collapse, technically known as apoplexy. And it is different from the course of symptom development where you see the gradual development between veraison and harvest of the leaf symptoms I showed you, and potentially the fruit spotting sometimes too. Instead, what happens is the entire canopy of the vine will wilt and die within 5 or 7 days. We as researchers don't know why ESCA switches from a more gradual to a more rapid course of symptom development. In California and in some other parts of the world, parts of Europe, for example, we found that bad apoplexy years seem to be associated consistently with a wet spring. And in California, the winter of 2016 and 2017 was our 4th rainiest on record. That made 2017 a really bad apoplexy year. Many, many reports were coming to me from growers all over the state about what's going on in my vineyard. The other thing that seems to be associated with it is prolonged periods of hot weather, 7, 10, 14 straight days of temperatures above 95 degrees Fahrenheit. And we usually start to see that happening in July here. The ESCA pathogens, Phaeoacremonium and Phaeomoniella, they sometimes team up with other diseases. One of them is called Blackfoot disease. It used to be called cylindrocarpon root rot. And together, these can cause a disease called young vine decline. This is a picture of a Grenache vineyard. It's 3 years old. I visited it this past summer. This vine here, some of them will be very, very stunted and low in vigor, and they'll never grow out of it. Others in the vineyard will look fine. The symptoms on the leaves can look perfectly green and healthy. Sometimes they'll have some amount of scorching. Sometimes they'll have a bit of discoloration. But if you look more closely and dig up these vines, you'll find that they have very few fine roots. So that's mainly the impact of the Blackfoot disease or the cylindrocarpon root rot. If you cut into the trunk of those vines, you'll find not just the black spotting and kind of rotten core at the center of that vine, but those spots are oozing black. And this is called black goo symptom. This is something that was recognized starting in California in the mid to late 1990s. If you look down at the base of the trunk and at the root crown in longitudinal section, and this is a sample of Pinot Noir from Oregon, you'll see this black center of the vine. This is all dead. This is not conducting anything for the vine. It's just this bit of healthy tissue on both sides. You can imagine if a vine starts out like this in year 3, it's probably not going to do well for years to come. And indeed, we see symptoms of young vine decline in mature vineyards. They don't grow out of it in spite of the name, young vine decline. This is a 15-year-old Zinfandel vineyard. The leaves don't have the typical symptoms of ESCA. There's certainly some scorching, there's some yellowing, but the fruit, no matter how much cluster thinning you do and shoot thinning, always is shriveled. And even with, adequate to more than adequate irrigation. Trunk pathogens have been identified in Oregon vineyards, and I've listed some here for Botryopspheriae and Eutypa diebacks, and also the ESCA pathogens have been found in Oregon. Achala, who's at Southern Oregon, she's done some work on trunk diseases with a team of other Oregon researchers, as has Jose, who's in BC. And then we've processed a fair amount of samples, and then every time I visit Oregon, I try to look around for different trunk diseases. The pathogens that cause trunk diseases all spread by spores, and rain is important for their production. That said, we don't know much about the conditions beyond that that make those spores germinate or infect the vines. We just know the rain is important. And based on our spore trapping studies, we know that the conditions with each rain event throughout the dormant season tend to be ideal for different trunk pathogens. It's not as if they all make their spores in December and then by the time March rolls around, they're done. It's like by the time March rolls around, there's different trunk pathogens that have the proper day length or the proper air temperature, or the proper amount of rain to make their spores. Every rain event is potentially a risky time in terms of sporulation for these pathogens. Once those spores are produced, depending on the species of fungus, they can be spread in different ways. For example, the Botryopspheridae dieback pathogens mostly spread by rain splash. That means the rain has to hit one of the small spore producing structures on an infected vine. And then as that rain droplet splashes away from where it hit, it carries the spores, which are formed in a sticky mass. And wherever that rain droplet lands, those spores stick to the surface. Eutypa dieback pathogens are all spread, their spores are spread by wind. So they need rain to be released into the air, and then the wind blows them around. So they can spread greater distances than, say, the Botryopspheridae dieback pathogens. The ESCA pathogens, some of them have spores that are spread by rain, others have spores that are spread by wind, and others have spores we found in the soil. They originate from grapevines that have symptoms, others that are still asymptomatic. Some of the same pathogens also attack different tree crops and native trees. And also, vines can get infected in nursery mother blocks. Sticky spores can land on the surface of canes, and when those canes are cut up into cuttings and grafted into bench grafts, that's one way that nursery stock can get infected. Also, the graft union itself is a wound, and these are wound pathogens. So some of the pathogens, and namely the ESCA pathogens, have been detected at the graft union. That said, we don't know which of these means of spread, whether it's by spore production and spore dispersal or nursery stock, or the actual sources where these pathogens may originate. We don't know which one always contributes more or less to disease spread for the different trunk diseases. These are just possible means of inoculum. Infection by trunk diseases is through wounds. These are what we call wound pathogens. They can't get in unless you cut into the vine. And one means of wounding vines, which happens every year, is by pruning. This is something that happens, trunk diseases are widespread worldwide. The diseases that are present in Oregon, California, Europe, it's more or less the same pathogens. And much of the work that's been done on how they attack vines has focused on pruning wounds. That said, other types of wounds are known to be entry points for trunk pathogens. In California, we have a lot of highly mechanized vineyards, and mechanical harvesters, mechanical pruners, any sort of mechanized machinery that goes through the vineyard can damage the permanent woody structure of the vine, can create wounds that can be entry points. No matter how sharp those blades are or how hard you work to prevent the shaker pads from, damaging the trunk, they do damage them to some extent, and the pathogens can potentially get in. Winter injury in cold areas, east of the Mississippi, in BC, in Washington State, and also in Oregon, is associated with trunk disease infections. Even in Texas, colleagues I work with there, in 3 to 5 years following massive winter kill events, there will often be major trunk disease outbreaks. And sometimes when training systems go about major changes in the vineyard and large cuts are made to the vine in order to do that, you can sometimes find wood cankers and wood symptoms associated with those entry points too. What we know about pruning wound susceptibility in grapevines is pretty limited. I've listed the very few studies that have been controlled inoculation studies, granted, but this is pretty much it. There isn't a lot that we know. And again, as I said earlier, most of the work that's been done on wounding in vines and how that impacts trunk diseases focuses on pruning wounds. That said, some observations have been made about pruning wound susceptibility, and this is where the management recommendations come from. For the Botrytis and Eutypa dieback pathogens that we have in California, several studies have shown that wounds are susceptible for a long period of time, up to a month, if vines are pruned in December. Whereas if you prune them in March, those wounds may heal up in only a day or two. And so the recommendation for how to manage based on those types of observations is to delay pruning to as late in the dormant season as possible. And then another possible option is to use a modification of delayed pruning called double pruning, which I'll show you a picture of in a minute. There's some conflicting results, though, about pruning wound susceptibility for some of these same pathogens that comes from South Australia and New South Wales. And it suggests that the wounds will heal at some point between 1 and 3 weeks, depending on the pathogen. And it doesn't matter when you prune during the winter time. It can be early, it can be late. That period of susceptibility is about the same. And so the recommendation that logically comes from that is that you should put on a pruning wound protectant, no matter when you prune, and expect it to work similarly. And I don't know how to apply these results to Oregon. I'm just giving you the information that we have. ESCA pathogens are quite different. There's been much less work done with them in controlled inoculation studies when it comes to pruning wounds. And we know from like one study in California on some of the ESCA pathogens that was in one vineyard and not over multiple years, that it seems the wounds may be susceptible for up to 2 months. That's consistent with what we've seen in studies we've done on protectants in our own lab, and also studies that have been done on delayed pruning overseas, showing that these two approaches don't seem to be very effective against ESCA. This is the technique I was mentioning earlier called double pruning, and it's a form of delayed pruning that, obviously we can't all prune our vineyards in March. There's not enough hands available in the Western United States to accomplish that. And so some growers will do what we call a pre-prune. They'll prune with a mechanical pruner or by hand, prune away most of the vine canopy, leave canes only maybe 10 inches long. And then come March, when the risk of infection seems to be lower, they'll make the final pruning cuts by hand. And in that way, that piece of cane you then remove with the second pruning pass takes away any infections that may have happened after the first one. There are pruning wound protectants. There are fungicides that are labeled for dormant season applications. And these are ones that have been shown in a number of studies to be effective against a broad range of pathogens. Topsin M and Pristine have been shown to be effective against some of the trunk pathogens, as has boric acid paste. The problem with these top two is they're not effective for very long. Even if there's no rain, they may only be effective for about a week. Here I've listed some of the organic materials that are obviously not fungicides. Vitaseal is something that hasn't been tested as much as the fungicides, but it offers the promise of being more durable. And if it does turn out to be that it is and provides a nice barrier, I think that that would be great. B-Lock does seem to be relatively effective against a broad range of pathogens. It can't be sprayed. You have to apply it by hand. And then I just want to mention briefly the biological materials. They haven't undergone as much testing in North America as the fungicides, but they do offer promise. and especially for organic vineyards. The thing is, you can't just swap them out with a fungicide, put them on once before rain, and expect them to work. They might need multiple applications to get established. And then lastly, I want to mention that a number of studies have been done, not with controlled inoculations, but tracking disease progression over many years, showing that the way you prune and train your vine does affect symptom severity when it comes to some trunk diseases. And we think that this might be the case because certainly the way you prune and train your vines impacts the amount of wounding that they undergo over their lifespan. And it's important to minimize the risk of infection by having as few wounds as possible, making those cuts small, and not having them on the trunk of the vine. Also, it's important to maximize the continuity of the connection, the vascular connection between the roots and the shoots. Wood is lost to trunk pathogens. Although those cankers and black spots and the decay are relatively localized, the vine literally has to work around them. Wood is also lost to the natural healing process. And so these two things combine to counteract the function of the vascular system over time. And with that, I'd like to say thanks so much for listening.
Thank you, everybody, for the opportunity to present today. My name is Mia Fisher. I work with Simoniten Sier here in the USA as well as Australia. Today, I'll be sharing a little bit more information about sustainable vine architecture. Our company has been dealing with vine structural development for over 30 years. The company was started by Marco Simonit and Pier Paolo Sir. Subsequently, we've been also doing private research, working with universities as well as other institutes. And for the last 20 years, doing vine structural development training across the world on different training systems, cultivars, and in different climatic conditions. Our expertise is sharing our know-how in the vineyard with regards to vine structural development. And this is done both in winter and spring pruning and in-field training. Just to share with you some of the countries that we work with and in across the world. And now I'll share with you a little bit more information about what we have observed over the last 30 years. The main source of our work is that we have seen that there are certain trends that are established in what I would call current and traditional pruning practices. Today, I'll be talking mainly about cane pruning, as I believe in Oregon, this is one of the main training techniques utilized. What we see is that one of those pruning styles utilized is known as the return cut. In this particular case, the vine outgrows the vertical space allocated to it, and ultimately the pruner leaves a spur position on the trunk structure to bring the plant structure back. As we can see here, the cane is now becoming difficult to wrap. So the idea is to control the growth of the plant over time. A big return cut is made. The proximity of this big return cut is completely dependent on the practices of the winery and vineyard. But what is important to note is the big surface area exposed to external pathogens. And if we take into consideration what Kendra just shared with us, this is a very important point. Over time, many return cuts are made to control the growth of the plant, both due to vertical or horizontal space available. And what we start to see is in our line of work that where we had desired a vineyard of uniform structure, we start to see heterogeneity or non-homogeneous vine structural development in the trunk structure. So we see big damage done to the primary structure of the plant. So that was the common return cut that is utilized to control the plant's growth over time in traditional vine structural development. Now I'll share with you a different method known as head pruning or crowning, or alternatively, many localized cuts in one area. Here we can see the selection of the fruiting cane is generally done rather randomly and is based on direction, vigor, and the ease of wrapping of the cane. Originally, when the plant structure is quite young, there are several solutions, but what we see is that over time, we make many cuts to the central crown of the plant, to the secondary structure, is what we would call it, of the plant. And over time, we start to see that the options that are available for the pruner become less over time. And as the vine ages, and this is a great example, this is possibly a bilateral plant, so it had two exits. We are now sitting only with one solution available. And eventually, finally, we have to retrunk the plant structure. So these are two methods utilized in standard pruning practices to control the growth of the plant over time. What I hadn't mentioned prior is that a large part of the work that we do actually has to do with vine dissections. What we have seen now together is ultimately the effect of the external impact of pruning on the plant, but through doing vine dissections, and we've done hundreds of thousands across the world, we have been able to understand the impact of annual pruning on the internal structure of the plant. Here we have a good example of the different impacts of pruning to the plant structure. In the bottom left-hand side, here we can see the effect of crowning, many localized cuts in one area, and we end up with an accumulation of deadwood, shown here as brown in the picture. And we can understand better why we started losing solutions in the central head where we are wanting to obtain our fruiting cane. Another type of cut, the big return cuts, ultimately results in desiccation down one side of the plant. And finally, the other situation is that through these big pruning wounds or an accumulation of cuts, we actually introduce and invite trunk disease into the internal structure of the plant. So annual pruning creates desiccation cones within the plant structure. This is a natural defense mechanism of the plant. And ultimately, where these desiccation cones occur and how aggressive they are to the plant structure is completely dependent on the pruning strategy of the pruner. Here we can see a good example of a big return cut made to the plant structure, as well as the effect of many localized cuts. These are two cuts made on the plant structure, mainly being a flush cut where we remove the basal buds, and this is a basal bud cut. But ultimately, a very important point is where we create many wounds on the plant structure, we ultimately result in one large desiccation cone. So the quality and quantity of desiccation within the plant structure is completely dependent on the pruner, but also the types of cuts made to the plant structure have the potential to increase susceptibility of pathogens entering the internal structure of the vine. So what we see over time is that ultimately traditional pruning results in the evolution of deadwood. This is architectural pruning, and over time, the living wood percentage within the plant structure decreases. This is one of these studies that we have done, and touching on what Kendra had shared with us, where especially in Europe, ESCA is quite an aggressive trunk disease. We have found that where we build up more living wood within the plant structure, the plant is more resistant to the influence and the development of ESCA within the plant. So living wood is a key component to supporting the vine structure. So traditional pruning consequences on the vine result in an accumulation of dead wood vessels, reducing the percentage of living wood, which we saw in the previous study is actually very important to protecting the plant against trunk disease. But living wood is also our storage reserve. And if we consider what is happening globally, we need the plant to have more living wood within its plant structure it helps us to deal with climatic conditions such as heat stress in a better way. But as we've also seen, or we'll see, bigger the pruning wounds, the greater the risk is for pathogens to enter the internal structure of the vine. And the canopy, we also see heterogeneity, deterioration, and finally premature decline of the vine. So 3 examples of the main effects of pruning are heterogeneity in the canopy, where we see differential cane health and length. Ideally, we are wanting the same diameter and length of our canes throughout the area that is our productive zone. Another result is cavitation and embolism. This is where the whole plant structure collapses in heat stress, which is this case. But on the topic of today, the introduction of trunk disease into the internal structure of the plant. In this vine, we can see that a big return cut was made. The pathogen entered the internal structure of the plant, thrived on the living wood inside, and continues to spread within the plant structure. This is a cordon, but it's a great example of how a big return cut was made, and either the pathogen entered through this wound or alternatively through other wounds. But it's good to see how this trunk disease has actually entered down into the primary structure of the plant, completely compromising the whole vine. And these, again, are only decisions made with the know-how that pruners have at present. I'll briefly share a little bit of information about ESCA that we have seen in Europe, mainly France, but this is just to show you a healthy vine on the left-hand side and a symptomatic plant similar to the symptoms that Kendra had shared earlier on. And this is a study that was done to show the effect that ESCA has on wine quality. But ultimately, we have found in our line of work that through doing cross sections of vines, that where we have created a big wound to the plant structure, the pathogen enters and then thrives on the moist wood and continues throughout the plant structure. Another part of the work that we do beyond training and consultation is also the handling of specifically ESCA as a trunk disease through curettage. But interestingly so, just to look at the impact that trunk disease has, this is a company that we've been working with, Bella Vista, that in 2006, we implemented the Simonite and Cyr technique. And out of interest's sake, it is very good to see that where we started to increase the percentage of living wood in the plant structure, moving away from big cuts, the symptoms or the percentage of plant loss due to ESCA reduced drastically over time. They have a 180 hectare property, so this was a considerable benefit to them. But over and above trunk disease, we can see that pruning wounds do affect the economic as well as the productive and qualitative capacity of a vineyard. So to share a little bit more about our methodology, we accept that the vine is a creeper and its desires to grow into the space available for it. A vine can also age. I believe that here in Oregon, you do have vines that have reached the age of 60 years old. So the potential is there for vines to age here. And where we allow the plant to grow, we can see that a vineyard can age endlessly. This is a 70-year-old vineyard. Obviously, we cannot take a bush vine structure and employ it everywhere. Our methodology ultimately looks at utilizing this respectful pruning technique and incorporating it both in cane and cordon. So our primary principle is to utilize branching, control branching, and utilizing cut separation. So respecting the chronological sequence of the vine. And doing so over the plant structure, allowing it to grow into the space available to it. Additional to that, we separate the cuts, making smaller wounds on the plant structure, therefore reducing its susceptibility to external pathogens, but also separating the area of desiccation to the area of which living wood can grow in the future of the plant structure, preserving the sap flow and the vascular flow of the plant, building up more living wood in the plant structure. This is a great example of a head pruned vine that was recovered, a great example of how we redirected and reestablished healthy living wood within the plant structure. Of course, in this particular case, this is more desiccation than trunk disease, but the rehabilitation is possible. And this is another good example of crowning, where many localized cuts were made, but we reestablished 2 channels on the side of the plant. So moving away from cross cuts and starting to ensure uninterrupted sap flow in the plant structure. Here again, this is again a good example of where we move away from cross cuts, big wounds, introducing trunk disease into the internal structure, and start to control on the left-hand side, making smaller cuts. We can ensure that we reduce the percentage of dead wood and or potentially trunk disease within the plant structure and increase the percentage of living wood within the plant structure on the left-hand side of the spine, and also over time. So moving away from big cuts, focusing on making smaller cuts in our annual pruning, working more with 1 or 2-year-old wood, we are reducing the potential of wound exposure and thus trunk disease, and also utilizing another tool of the vine's own defense mechanism, namely spare wood. And this is a good example of the utilization of protective wood, so that we can start moving towards an evolution of living wood within the plant structure, as this will support the vine in many years to come and is more a part of sustainable vine structuring. So here, this is more respectful of the physiological growth of the plant instead of architectural pruning as we saw before. So redefining our logic, looking at the space available both horizontally and vertically to the plant structure allows us to build up a stronger vine and therefore a plant that can sustain the end of time. Ensuring a healthy plant structure with much living wood, we can also ensure that we end up with a more uniform ripening of our vines. So Simon Eaton-Sear's main objective is through prevention, we can establish longer lived vineyards. Thank you very much.
So thank you, everyone. It was great. I think we learned a lot. And I think also that we have to be aware that it's quite scary. In our vineyard, personally in Oregon, we don't really see yet the symptoms that have been presented. But I think we have it clearly, as Kendra was showing. But anyway, I have a couple of questions that came up. We discussed in the past a little bit during our pre pre-meeting that there is quite a bit of vineyards that's trying to re-trunk vineyards and to also like selfishly sometimes we are at Lingua Franca trying to overgraft. And so question to you too is how do you think that can impact the future of trunk disease, and do we have to be extremely careful, and do you think it's a good a good option and solution?
Good question. Mia, do you want to go first?
I can. In our line of work, of course, if it is needed to be done, it's always about good practice. If you have to make a big wound to the plant structure, either because you are regrafting because of a change in clonal or cultivar, or because of decline in the vine structure, we need to implement it in a good way. So utilize your applicants, your wound sealants, because you are making a large wound to the plant structure, ensuring that you do that at a right time, and also ensuring that you leave enough space for the desiccation to occur into the plant structure, because these are 2 separate things. Desiccation is normal with year-on-year pruning. Trunk disease is a separate introduction to the plant's internal deterioration. So these are two separate things. So apply the wound sealant, of course, for the trunk disease, but leave space for desiccation to occur.
And Mia, in school, we always talk about having this rule of thumb saying the cut needs to be twice the cut length needs to be twice the size of the diameter. Does it make sense to you, or is that something good for the industry? Or
it depends on the age of the wood. It really does depend on the age of the wood. In our line of work, we are always respectful when we enter a new area. So we'd rather say keep twice the diameter on younger wood. If you're cutting into a trunk structure, I would say rather leave more than that, if you can. Obviously, if you're seeing signs of trunk disease, you have to go lower, but ideally keep more. Not too much, of course, as well. About 3 times or 4 times the diameter of the cut.
And I would just add to what Mia said that, retrunking is a real drastic measure. It is, the last thing you want to do before you replant the entire vineyard, because it is so heavily impacted. The promise it offers, though, is that you're assuming you can cut away all the infected wood with the cankers that have been building up in the above ground part of the vine. That's what you want to do. The problem is if those infections are in the trunk, there's really no way to cut them out. And so I uploaded to with our presentation material a poster that we made for Spanish speaking farm workers along these lines of like, don't wait until the vine looks almost dead to retrunk it. And in our economic simulations we have done with an economist at Cal State Sacramento, his name is Jonathan Kaplan, he found that, and granted, these weren't long-term studies in the vineyard, but they were long-term economic simulations, that waiting until you're 15 or later is not economically feasible if you're going to retrunk a vineyard. You really need to do it sooner rather than later from an economic perspective. The idea being that if you've lost yields for years and years and years, and then you decide to retrunk the vine, you take it out of production for 2 years, you have to pay someone to cut it out, tear away the canopy from the trellis system, and retrain it. And so it's a big decision to make. And then you're hoping that it'll turn into a healthy vine afterwards. And that's a big risk. And as Mia said, you have to protect those cuts. It's a huge cut to the trunk. We ideally recommend that people do this during the growing season because vines tend to heal more rapidly when they're actively growing than during the dormant season. And then also if there's no rain during your growing season, then that's also a good reason to do it then. The one thing I just want to mention about top working or regrafting, some growers do this with material that they get from their own vineyard that they can test and try to ensure that it's healthy. And I would say that, if you're getting material from other vineyards or from nurseries, probably you want to be careful about its quality. If you're trying to rework a vineyard that has disease in it, you don't want to bring in more disease material on top of that, whether it's trunk pathogens in it or viruses or anything else. So anytime you bring 2 bits of plant material together, you're doubling your risk of there being a pathogen in there.
Absolutely.
Great, thank you. So I just 2 questions came to my mind since we were discussing and with your presentation. One for me, one of my challenge as I was trying to bring this concept a little bit that I've learned in France about Guyot Poussard and this technique was the fear of the people to have an extended, space in the middle. And some of your picture showed it, and I was just wondering if you could explain a little bit more of what you think.
So in our line of work, it's always about preservation of the vine structure. So accumulating living wood and not growing too quickly. So that's that whole concept of controlled branching. But of course, that can result so that the plant grows oblique and the two positions move away from each other. In cane pruning, we have to remember that the cane is flexible. You can do anything with the cane. So if the fear is that you've outgrown that space, there's nothing stopping you from bending it back. If that explains it. If we think about vine structure development, ultimately we have to take into consideration the space that the plant is going to grow into over the next 40 to 50 years and move away from only thinking about where it's going to grow into in the next 10, 15 years. Because ultimately, if we don't give the plant the space to grow into over time, you'll be making the big cuts or aggressive cuts to the structure, which will force you to have to retrunk in the end because you've introduced those pathogens. So to answer your question, the cane is flexible. So
Makes sense. And also, it takes time to grow. I mean, if you the cut in the right direction or a small cut, then you should take quite a bit of time to get to this.
Absolutely, absolutely, and there are many components to that. Where do we start our secondary structural formation? What's our starting point? Where we're growing to? And that has to do a lot with fine structural vision. And it's something that I encourage and employ a lot of people who are planting young vines to think about where they want their vine to be in 50 years, because that would change or reshape the whole way they approach their training system and their vineyard. Because the objective there is to have their vines in the ground 50, 60 years down the line and still have the original primary trunk structure.
Thank you.
Okay.
Okay, well, time to wrap up, but the last question open is for me. Wanted to ask Kendra really quick, California becoming worse and worse in trunk disease to your research? Sorry for the quick.
I think that vineyards that are kind of between the ages of 15 and 20, which we have many of. We had a big expansion of vineyard planting in the mid to late 90s and then in the 2000s. And those vineyards are being replanted in large part because of trunk diseases, but also viruses. When you see vineyards of those ages, it's rare to see just a virus or just one trunk disease. The two kind of go hand in hand. And yeah, we have a lot of replanting going on because of both of those things. In fact, trunk diseases and viruses are the top main reasons that our vineyards are being replanted here.
All right, thank you. So we have a lot of work here and we have to be careful in Oregon. Thanks, everyone. It was great. Thanks for your time and I learned a lot. So I'm very happy we did that.
Thank you.
Thank you. It's been great.